Welcome, everyone, and thank you for joining us today for 3 tips to simplify 3 procedures in the IVF lab. Today's webinar will be presented by Dr. Joseph Conaghan. Dr. Conaghan has over 2 decades of experience in human embryology and is best known for his studies on embryo culture and metabolism. He trained in embryology and andrology at the Hammersmith Hospital in London and graduated with a PhD in human embryology from the Royal Postgraduate Medical School at the University of London in 1994. He has been the lab director at Pacific Fertility Center in San Francisco in California since 1999. He's a strong advocate for elective single embryo transfer in IVF and therefore for high standards for culturing and vitrifying embryos. With that, I will hand it over to Dr. Conaghan to begin his webinar.
Okay, thank you, Trigera, for that kind introduction. Can you hear me okay?
Yes, I can.
All right, well, welcome to all of you who have joined our webinar this morning. It is 9 a.m. here in San Francisco, and as you're probably aware, we have a lot of fires burning in California, but the sun is up today and the sky is blue, so it's a great day. It's morning time here, so I have my coffee and we are all ready to go. So I am going to cover 3 topics today just to mix it up a little bit. I'm going to talk a little bit about the culture environment in the IVF lab. I'm going to talk about vitrification, and I'm going to talk about micromanipulation. Now, my sort of thought behind doing this webinar today was that I've been an embryologist for a long time, and I really enjoy being an embryologist. I enjoy being in the lab. I like being in the lab every day, even though I'm a lab director. And I think for some of us who've been doing this a long time, there's probably sort of very small tips and tricks, little things that we do every day that you don't necessarily pick up as a young embryologist very easily, because a lot of these things are very subtle. So I'm hoping that we have a lot of young or new embryologists on the call today. I'm sure we have people like myself, older lab director, senior embryologist too. And I've tried to throw in a little bit of something for everybody here. So some of the things I'm going to be talking about are incredibly simple, and some are a little bit more complicated, but they're all designed to be things that you can leave this webinar and put into play right away in your IVF lab. I love sort of talking about practical stuff that, you know, you can come away from a seminar, you can come away from a webinar, and you can actually do these things immediately. So with that, let's start with embryo culture and see, are there things that we can do in the IVF lab to simplify and streamline this process? So one of the things that sort of, I don't know, catches my eye maybe when I visit different IVF labs around the world is that a lot of people are still using big box incubators. We are here in San Francisco. We still have a lot of big box incubators. And these incubators use a lot of nitrogen gas to depress the oxygen levels inside the chamber. And it's always kind of strange to me when I go to some big labs in different parts of the world and I see the way that people are using their nitrogen gas. So that's why this is the sort of first topic this morning, because this is something that sort of bothers me a little bit. And I'm like, why don't more people do this? Right? So if you're using big box incubators in your lab, the chamber in those incubators is usually somewhere in the range of 100 to 150 liters, right? Relatively large chamber. And if you're pumping in nitrogen gas to depress oxygen levels, you're usually using, you know, more than 100 liters of nitrogen gas in the chamber at one time. So you're using a lot of nitrogen gas, right? And in the very beginning, when we first started doing low oxygen culture, we used these bottles of compressed nitrogen gas, right? And you can connect these up to your incubators and you can pump in the gas. And because these bottles contain, you know, a very limited amount of gas, you end up having to change these cylinders very often. Trigera and I were in a lab in Sweden back in February of this year, just before the pandemic hit. And we visited this lab, and they had a lot of big box incubators, and they were using these compressed gas cylinders to get their nitrogen. And they were changing these all the time. Every day they had to change these cylinders. And it was a real pain for the embryologist to have to do this all the time, because these incubators, these big incubators, they use a lot of nitrogen gas. Now, there is an alternative. And for many of you, you already know this, particularly people working in the US. I think this is very common, but in other parts of the world, maybe not so common. If you look at your big cylinder that you use for your liquid nitrogen supply, right, you have a valve on here where you draw off your liquid. That's shown in this left panel right here. But here's a top view of a tank. And this tank is what we call a high pressure tank. So this is not one that you would typically draw liquid from. This is one that's at a higher pressure. And over here on the right-hand side, you can see on this little ventilation valve right here, the pressure on this tank is 230 psi, right, pounds per square inch. And you can see that on the little gauge right here. And there's the vent. If the pressure gets higher than it will vent off, right? And there are numerous sort of outlets on this tank. So over here on the left-hand side, we have an outlet that allows you to take gas from the tank. And then over here, further left, we have an outlet that allows you to take liquid from the tank, right? So these tanks are very versatile. And what you can do is, instead of connecting your gauges or your hoses to a bottle of liquid, of compressed nitrogen gas, you can just connect it to the tank, to the liquid nitrogen tank. And the fitting is exactly the same. It's exactly the same size. It takes the same hose. It takes the same gauge. Whatever it is you use, you can just stop using these guys and you can transfer over to these guys. You can just connect up your hose, turn on the valve, and you're ready to go. Right? Now, the same hardware works, right? So I just showed you a hose, but here is a gauge right here. You see it's connected to the bottle right here. The same gauge you can connect to your liquid nitrogen tank. It's very easy, very simple. Right? If you do this, if you move from using bottled nitrogen gas to evaporated liquid, you will get about 15 times more nitrogen gas from this guy than you will get from this guy. The savings are huge, right? So if we think about that in practical terms, so here's our lab and here's our big box incubators right here. Got some bench tops down here, but we got big boxes right here. One of those cylinders of compressed nitrogen gas will give you enough nitrogen to fill one of these incubators 40 times, right? But if you have, let's say you have 5 of these incubators, now you only have enough gas to fill each one 8 times, right? So you'll go through those cylinders of gas very quickly. If you change to using a large cylinder of liquid nitrogen and use the evaporation, the gas that's evaporated, you will have enough gas to fill your incubators 1,500 times. So the difference is huge. The difference is really huge. Huge savings here, and you're using evaporated gas, right? It's distilled gas. It's probably the cleanest gas that you can get. So not only do you have savings in terms of efficiency and how often you have to change your tanks, but you're using probably the cleanest gas that you can get to feed your incubators. And remember, 89% of what's in your incubator, or 90% of what's in your incubator, is nitrogen. So the cleaner that nitrogen is, the better your culture environment is. All right, so that's tip number 1. Change from using compressed gas to liquid evaporation. Now, continuing with the theme of embryo culture, you see these Petri dishes that I have right here? See how pink they are? Well, in days gone by, we had phenol red in the medium, and as an embryologist, you could see pretty quickly whether or not your media was equilibrated. And for those of you who've been around the block like me a few times, you can tell right away that this kind of purplish color in these dishes means that this medium is not equilibrated, right? We're looking for more of a kind of a nice salmon pink color and not this dark purpley color that we see right here. Now, modern media are emitting phenol red, like it's not as common anymore as it used to be. So it's not so easy for the modern embryologist to know when their dish is gassed and when their dish is not gassed, when it's equilibrated, right? So the question that I have, and this is particularly for young embryologists, is like, when you make up a dish and you put your drops in the dish like this and then you cover it with oil, how long does it take for that dish to equilibrate inside the incubator? Right? If we put that inside the CO2 incubator and we don't open the door, how long does it take for that dish to equilibrate? And I've asked a lot of people this question, and I always get quite a variety of answers. Some people think it takes 4 hours. Some people think 6 hours. Some people think it takes 24 hours. Well, we've actually gone ahead and done the measurements, and when you take oil and media out of your refrigerator and you make up a dish, the first thing that happens is that the pH starts to rise. Because when you put this cold media into the incubator, CO2 that's already dissolved in the media comes out of solution as it warms up. And then eventually CO2 from the incubator starts going back into your dish and the pH starts to come down. And then this is hours down here on this axis. And you can see that maybe after about 8 hours, you're getting pretty close to where you want to be. We've got a target of 7.34 for our pH right here. So the minimum amount of time that you probably want to allow a dish to equilibrate is 8 hours. Definitely not less than 8 hours, right? That's your minimum. But really to get to full equilibration, you probably need a solid 12 hours, right? So you need a good 12 hours to equilibrate a dish. If you're really in a bind and you need to make up a dish and you don't have that much time, maybe you're okay after about 8 hours. You're getting pretty close and you're definitely in the physiological range. But the minimum is 8, and you probably need a good 12 or more hours to get full equilibration of that dish, right? So this is, again, for the young people out there who don't have a good feel for this, you cannot make up a dish of culture medium under oil and use it the same day in the IVF lab. If you come in the morning and discover that you've forgotten to make a dish, you cannot make one up and have it ready, you know, by lunchtime or later that same day. It takes longer than that for a dish to equilibrate. Now, once the dish is equilibrated and you take it out of the incubator, how quickly does the CO2 come back out of solution? How quickly do you have to work with that dish so that you don't affect the pH of the medium and stress out your embryos? Well, we tried to measure this, and we got dishes and we had them fully equilibrated, and we took them in and out of the incubator, and we were unable to measure any changes in pH when we took a fully equilibrated dish out of the incubator for less than 10 minutes. Right, so if we took a dish out for 5 minutes and put it back in, because the oil really holds the CO2 in there really well, we were unable to measure any changes. And it wasn't until we took a dish out of the incubator for a full 10 minutes that we were able to measure a change. And that's what you're seeing on this graph right here. So at the beginning, the dish was equilibrated to a pH somewhere between 7.32 and 7.34. And we took it out of the incubator. This little flat area on the graph right here represents the time that we took it out of the incubator. And when we put it back in the incubator after being out for 10 minutes, the pH was relatively stable for a little while. But then even though it was back in the incubator, the pH went up a little bit and then eventually came back down. Took about 4 hours to get back to where we were at our starting point. Well, somewhere between 2 and 4 hours. But you can see that the pH pretty much stayed within the physiological range. It went up a little bit, but not drastically. And this was after 10 minutes out of the incubator on a warm stage. So the message here is that if you take an equilibrated culture dish, remember it took 8 hours minimum to equilibrate it in the first place, but really 12 is needed. And if you then take that dish out of the CO2 incubator, you have up to 10 minutes to work with that dish before the pH will measurably change. So everything that you're doing in the IVF lab, if you're taking a dish out to score your embryos, if you're taking a dish out to do some assisted hatching, anything that you're doing with an equilibrated culture dish that has eggs or embryos in it, you have 10 minutes, right? That's your sort of timer that you should be thinking about mentally in your head. I can work with this dish for 10 minutes, put it back inside the incubator, and the pH of the medium will not measurably change. All right, that's embryo culture. So we're through section 1 of this little presentation this morning. So we've talked a little bit about the nitrogen gas and helping to use your incubators more efficiently. We've talked about the equilibration of culture media and how long you can handle your dishes outside the incubator. Second topic, now I want to talk a little bit about vitrification. And those of you who know me know that I am really passionate about this. This is a subject that's really close to my heart. And first thing about vitrification, and again, you know, I visit a lot of labs around the world and I see people do these things in different ways. And I would say that in your lab, you should really work hard to set up your work area so that it's really efficient and it's designed to allow you to do your vitrification and warming procedures really efficiently. Now, I'm going to show you a little video right here in a second, but just want to point out the features here, right? So here's my work area right here. So I'm sitting at a microscope. It's my work area, and I've actually got a heated stage right here. So this work area is warm. It's at 37 degrees centigrade. I have a little warming incubator right here, right next to my work area, right? And then right here, I have my liquid nitrogen bath. Right? So I've got this sort of little triangle going where I have everything that I need within arm's reach. So I have a cryolock right here that I'm going to warm. Right? It's sitting in the liquid nitrogen bath and it's ready to go. In here, I have a dish of warm thaw solution, and that's been warming in the incubator for at least an hour. So I know that that's at 37 degrees. And then I have my microscope here with my warm stage. So I'm ready to do my warming event. Now, hopefully this video will come across on the webinar this morning. Sometimes these videos can be a little stuttery or they can be a little glitchy. So you get out your warm plate of media and you very quickly grab your cryolock and you pull off the cap of the cryolock and you get it into the warm media like instantly, right? So by having this little triangle right here, everything is within arm's reach, everything is really close. You can get your cryolock with your eggs or embryos on here out of the liquid nitrogen and into a dish that's at 37 degrees without any loss of temperature. And by doing that all very quickly and having a very efficient workspace, you're getting the maximum possible warming rate for whatever tissue that you have on that device. So set up your area to be efficient like this, have it nice and clean like this, and don't have it all cluttered with stuff in your way, and you will get the best warming rates that you can. All right, next tip that I have for you, and again, this is something very simple. When you are doing a vitrification or a warming procedure, you should lubricate your pipette before you start doing anything, right? Now, what do I mean by lubricate your pipette? So in this picture that you see right here, I'm actually about to start an egg vitrification procedure. So I've made a drop of my handling medium, I have a drop of equilibration solution right here, and I have a drop of equilibration solution right here. And in the egg vitrification procedure, we're going to be merging these drops together, right? But before I pick up my eggs, before I go to the dish where the eggs are located, I will bring my little pipette, my little stripper tip right here, and I will go into this medium, this handling medium, and I will lubricate, I will preload my pipette with medium right here. Now, what medium am I using? Well, for vitrification, we need an equilibration solution and we need a vitrification solution. But the wash solution that comes with your vitrification and warming kits is the one that I use to make this drop right here. Right? And that's the one that we're going to lubricate the pipette with. Now, why do I do that? Well, these solutions, actually all three of these solutions contain 20% protein. Most of the culture media and products that you use in your IVF lab only contain 5 to 10% protein, but these guys right here all contain 20% protein, right? And the reason they contain 20% protein is because the protein protects the eggs or embryos from osmotic shock during vitrification and warming procedures, right? It helps to sort of modulate the egg or embryo's response to these sort of difficult osmotic conditions that they're going through during vitrification and warming. So they contain 20% protein. So if you lubricate your pipette in here in this drop before you start, you have now sort of lined your pipette with this 20% protein solution. So your pipette's not going to be very sticky. And you know how sometimes we get little oil droplets and stuff inside our pipette, and they can sometimes get in our way and they can be a little bothersome. Well, if you lubricate your pipette with your 20% protein solution right here before you start, you will not get any of these oil droplets or anything inside your pipette, and things will go in and out of your pipette really smoothly, right? So before you start, get in the habit of lubricating your pipette from this drop, and then everything will go smoothly from there on in. Now, during the process of egg vitrification, we start off with some eggs that, look pretty normal size. And when we go into the equilibration solution, the first thing that happens is that the eggs shrink, right? Not very dramatically, but they do very much definitely shrink. And you can see that right here, most pronounced probably. And that happens relatively quickly because remember, water is a very small molecule and water will leave the cell almost instantly when you put these eggs into your equilibration solution. So the first thing that happens is that you see that they will shrink. And then as you leave them in the equilibration solution, the cryoprotectants will work their way into the eggs, and the eggs will gradually come back to normal size. So as you're doing an oocyte vitrification procedure, you should be sort of paying attention to the shrinkage first and then this re-expansion. Because at the end of your incubation period in the equilibration solution, you're going to be moving these guys into your vitrification solution, and you want to see that they've come back pretty much to the same size that they were when you started out, right? If you want to see that they're back to like 90 or 95% or 100% of their initial volume before you move them on to the vitrification solution. Now, I have a little video right here. This is a time-lapse video of an oocyte in the equilibration solution. And the water has already sort of instantly left this cell, but you can see now, as you know, this is a sped up video, it's time-lapse. You can see that the egg is coming back to its normal size. And this little video is just 20 seconds. But what we're looking at here took 10 minutes it was a 10-minute filming, but because it's time-lapse, it's only 20 seconds. So essentially every 2 seconds that you're seeing on the video right here represents 1 minute in real time. So you can see that the egg has come back to normal size. And in the equilibration solution, the egg shrinks and it re-expands, right? It shrinks one time and then it re-expands. Eggs have all cells, in fact, have limited tolerance for shrinking and swelling. They don't like shrinking and swelling and shrinking and swelling. They don't like to go through that process over and over. So this is actually the only time in the equilibration solution is the only time where you should have shrinking and swelling like this, right? Once you get back to full size like this, we're going to now go into the vitrification solution, and in the vitrification solution, we will shrink only, we will not swell, right? So we're ready to move forward into the vitrification solution. And, you know, if you have a lot of eggs, you might have more eggs in your equilibration solution than you're going to put onto one cryo device, right? So in this example right here, I have 4 eggs, and I'm going to put 2 onto each cryo device. So at the end of my incubation in the equilibration solution, I will come along and I'll look at these 4 eggs, and I will, for my first 2 eggs to be vitrified, I will look at these guys and I'll say, okay, who are the ones that are most re-expanded? Who are the ones that are closest to being back to normal size? So I would probably pick this guy over here on the right, and I'd probably pick this guy over here on the left, and I'll take those into the vitrification solution and get those guys vitrified, and then I'll come back for these last 2 right here. And by doing that, I'm taking the 2 eggs that I think are most ready to be vitrified. They're almost back to their normal size. And then the ones perhaps that are not fully back to normal size, like this guy and this guy, they get an extra minute in the equilibration solution just to sort of expand a little more and get back to their normal size. Right. Now, during the procedure, you will see here, I've got a little video here, and I'll play this video. You've got to see if my video is playing. All right. There we go. All right. A little glitchy, but we got it going. All right. Now, during these procedures, you want to make sure that you just make your drops as you go, right? So here's the first part of the process right here where we're merging the equilibration solution and the handling medium or the wash solution, right? You'll notice that the other drops, like the ES3 drop right here and the VS drops, they're not in the dish yet, right? We just make these drops as we need them because these drops are very small, and we don't want any evaporation as we're going along. I mean, small drops like these have a large surface area to volume ratio. They will evaporate really quickly, right? So we just make the drops that we need, and the ES3 drop and the VS drops, they will be made in due course, but we do not make those at the beginning of the process, right? We just make the drops that we need. We make them as we go along. We also have the lid of the Petri dish right here, and at the beginning of the process, we keep the lid on the Petri dish to minimize evaporation. And then, of course, whenever we're not actually working on the drops or merging or moving, we keep the dish off out of the light path of the microscope so that the eggs are not sitting in the light path all the time. Right. And then finally, we get to our vitrification solution. And your tip here is that you want to get in and out of the VS solution as quickly as possible. Right. Now, when you bring your eggs to the vitrification solution, there's a very dramatic shrinkage, right? See here how sort of ugly the egg looks as it goes into the vitrification solution, because this solution has 30% cryoprotectant and it contains some sucrose. So as soon as your eggs go into the vitrification solution, the water is really sucked right out of them. Right? And as soon as they shrink down to this size and the water is sucked right out, you are ready to move forward and get them into your liquid nitrogen as quickly as you can. You are not waiting in the vitrification solution for any re-expansion. Once they shrink down to, about, this is probably about 30 or 40% of their initial volume. Once they shrink down to this size, they are ready to be vitrified. You do not need to wait for any re-expansion. When the cell is shrunken like this, the cytoplasm is remarkably stable, and you should load it onto your device and plunge it into your liquid nitrogen in perhaps 30 to 60 seconds from when you first went into the vitrification solution. So this last step always has to happen really quickly and really efficiently, and you're not waiting for any re-expansion of these cells. As soon as they shrink down to their sort of minimal size, you can move forward and get them into the liquid nitrogen. Right. Now, here's a little dish that just shows this is the dish after I finish, right? So here's a drop of equilibration solution right here, and I like to have the eggs sit in there for about 10 minutes. And then when the eggs have shrunk and come back to normal size, we then go through these washes through vitrification solution. And you see how untidy this looks? You see all these little squirts of media right here? That's because when I put the eggs in this first drop of vitrification solution, I emptied out my pipette several times and went back in and refilled it so that I'm removing any trace of the equilibration solution that might have got carried over. And I do that in the second drop. I empty out my pipette and refill it here, and even in the third drop, so that the eggs in this last drop right here, there should be no trace of that equilibration solution that's come all the way through to here. Right? So it looks kind of messy and ugly here right at the end, but you can really tell just by looking at this that I really went to a lot of trouble to wash out my pipette and rinse it out as I was moving my eggs across to the last vitrification drop. And then once you get there, you are then ready to load your devices onto your cryolock or your cryo tip. And this is Gina in my lab. She's loading her eggs onto her cryolock right here. So she loads them right on. And remember, she's working in this nice efficient space. So her liquid nitrogen bath is right there. And she gets in and she stirs it in. And now your samples are vitrified, right? So having this nice efficient workspace, it allows for plunging of devices after loading like instantly, so you get a great cooling rate. There's no delay right here. And then on the warming end, same thing. You have your water, you have your liquid nitrogen bath right there and your microscope right there, so you can warm things really quickly, right? So set up your work area to be efficient and versatile and allow you to do what you need to do. All right, that's it for vitrification. We're all done talking about that. So we've talked about embryo culture, we've talked about vitrification, setting up your workspace, lubricating your pipette before you start and make your life so much easier. And then pay attention to the shrinking and swelling of your cells. Remember, cells have a limited tolerance for shrinking and swelling, so you don't want this to be going on all the time. Right? It happens only one time in the equilibration solution, and then when you get to the vitrification solution, you have shrinkage only, and then loading your device and quickly into the liquid nitrogen. Right? You do not need to hang around in the vitrification solution. If you're staying in the vitrification for longer than 1 minute, you're staying in there too long. All right, third section of my talk is going to be on micromanipulation. And the first thing we're going to talk about is ICSI. Now, some of these tips are really sort of geared towards the young embryologist or embryologists who are just learning ICSI or trying to get better at doing ICSI. Right? So the first thing that I try to impart on people when I'm teaching ICSI is that you've got to get a good hold on your oocyte before you come at it with a needle. Right? Your needle is going to be going into the oocyte, maybe even moving around a little bit inside the oocyte. You do not want the egg to move during the procedure. So you got to get kind of a death grip on the oocyte so that it doesn't move when you're doing your ICSI procedure. And one of the things that I'm very fussy about is, when I pick up my egg, so let's say egg right there, and here's my holding pipette right here. one of the things that I'm very fussy about is I pick it up, but then I also make sure that the egg is sitting on the bottom of the dish. You see this brown line down here? This is the bottom of the Petri dish. You don't want to do your injection with the egg sitting up here and not anchored on the bottom of the dish. Because when you push in the needle, the only thing that's holding the egg is the holding pipette. And if your angles aren't perfect here, or if you're moving around inside the egg can roll off the holding pipette to one side or another, right? But if you move the egg down so that the egg is actually touching the bottom of the Petri dish, you are now holding the egg here and you're anchored down here. So when you stick in your needle into the egg now has much more limited ways in which it can move. You've got sort of 2 points where you've really, you've got places where the egg really can't move, here and here. So your egg is now really stable and it should not move during the process, right? So here's an egg right here, and I'll show you this little video. And you'll see when we pick up the egg on the pipette, you see how it goes out of focus and then comes back in focus and goes down? When you bring it down onto the bottom of the dish, you see the egg roll a little bit. You bring it down. I see it roll right there. Now you know you're touching the bottom of the dish. So you're anchored right here on your holding pipette, and the egg is touching the bottom of the dish. So now when you come along with your ICSI needle, that egg is not going to move. It is very stable. All right. Now, just one last sort of small detail on that. You see this holding pipette right here? The suction is so much that you've actually distorted the egg a little bit here, right? You've really got a good death grip on the egg. I like that. I like having this really strong grip on the egg, and I like seeing this little bit of distortion right here, because that tells me that I know I have a good grip on the egg, right? So I actually do like seeing that little nipple form right there, because during my ICSI procedure, I know that my holding pipette has really got this death grip on the egg, and the egg is not going to move. So now that we've got this good grip on here and we're anchored on the bottom, our egg is, ready to be injected. So here's another guy right here. And now we're going to pick it up and we're going to do an injection. All right, so we get our good grip on the egg. We make sure we're touching the bottom right there. Got a good steady hold on the egg. The egg is definitely not going to move. So now we can bring in our sperm. Now I want you to watch in this video what happens to the sperm cell at the end of the injection. Right? We bring our sperm cell down through the ICSI pipette, coming down. All right, so in we go, and we push on the membrane. Boom, the membrane broke, the cytoplasm flows in. You see the way the sperm came out the end of the needle and cleared the end of the needle? Did you notice that? The sperm did not stop at the end of the needle. The sperm cleared the end of the needle and kept going past the end of the needle and passed into the cytoplasm. Right? Let's just watch that one more time. So watch this. The sperm does not stop at the end of the needle. The sperm keeps going. Okay, there's the break. Cytoplasm is flowing, and the sperm goes way beyond the end of the needle. So for young embryologists out there, if you really want to be really good at doing ICSI and get really high fertilization rates of 85 or 90% for each M2 that you inject, make sure that at the end of your procedure, the sperm has gone past the end of your needle. If you do an ICSI, and at the moment you're pushing the sperm into the egg, the sperm stops right there at the end of the needle, the egg membrane may have healed. That membrane repairs itself really quickly. And if your sperm cell stops right at the end of the needle, it's possible that the membrane has healed and you have not put the sperm into the egg. If that happens to you, pick up the sperm again and do the injection again. Right? Just take the sperm back and go back in and do the injection again. Do not stop until the sperm has gone past the end of the needle, because then you know that the sperm is definitely inside the egg, right? So always make sure with your ICSIs that you are confident that the sperm is definitely in the cytoplasm and that it has shot out of your needle and gone way past the tip of the needle, right? That might be the most important tip that I'll give you today. All right. Other micromanipulation procedure that I want to talk about, and this is what the last topic for me today, is tubing of cells after blastocyst biopsy. Now, this procedure, some of us think maybe it's a relatively simple procedure, but again, for young people or for new embryologists who are learning this, it can be quite tricky. And the objective here is to get a very small piece of tissue. Maybe you've got 4 or 5 cells and they're a little bit sticky. They've just been pulled off a blastocyst, and they're tiny and they can be hard to see. Your objective is to get them into this little Eppendorf tube here with perhaps 2 to 5 microliters of medium in the bottom. Right? And it's tricky. It's a little tricky thing to do. So are there things that we can do to make this simpler? So in this picture, I've just set up a little mock. This is not real embryos right here. I just set up some dishes just to show you this kind of process. So after we do an embryo biopsy, we have a dish over here where we did the biopsy, right? And in this situation, we have 4 embryos, 1, 2, 3, 4. So we biopsied 4 embryos in this dish. The embryos are back in the incubator now. They're back in culture. So what's left in each of these drops? In this drop right here, we have the cells that we biopsied from embryo number 1. In this drop right here, we have the cells that we biopsied from embryo number 2, and so on. Right? So that's the dish right there on the left where we did the embryo biopsy. Now, we got to get the cells from each of these drops out of there, and we got to wash them in buffer before we put them into an Eppendorf tube, right? Because we want to get rid of anything else that might carry over from this drop. So we're going to wash the biopsied cells 3 or 4 times in this buffer solution, and then we're going to put it in the Eppendorf tube, right? And we got to do that for all 4 of these embryos right here. So what I like to do is I like to take one pipette and I like to move these guys across 1, 2, 3, 4 to the first row of drops right here. Right? That's my first step. And once they're across, I will change my pipette tip and then I'll wash each one in turn. So here's my tip for you. In this dish where we're going to be doing the washing, put a drop of PVP in here. See that right here? Here's my PVP. So put a drop of PVP in there. And before you start moving the cells, lubricate your pipette with the PVP. Remember, these cells are going to be a little bit sticky. You've got to go into the dish where you did the biopsy. So there's oil in that dish, right? So you've got the danger of getting oil in your pipette. You've got the danger of the stickiness of the cells. But if you lubricate your pipette in PVP before you start, you will eliminate any complications from stickiness or from oil in your pipette. Just like we did with vitrification where I said to lubricate your pipette before you start, same here. But this time, if you use PVP, it will really lubricate your pipette and your cells will not stick to your pipette. Now, use the smallest pipette tip that you have. In our lab, we use a variety of pipette tips, right? We use 170s, we use 130s. These 130s are the smallest ones we have, so that's the pipette tip that I like to use for transferring the cells. Right? So here's your procedure. When you start, rinse your pipette tip in PVP, and then go to the biopsy dish and transfer all of your cells over to the wash dish. Right? So if I have 4 of them, as in this example right here, transfer number 1, rinse your pipette in PVP. Transfer 2, rinse your pipette in PVP. 3, 4, same way. Right? Now, if you look into these individual drops right here, after the cells have been transferred, and it's hard to see in this picture, but there's a tiny little speck right there. That's the cells. Right? And you'll notice up here that there's a little bit of an oil slick on top of the drop. Right? So because of that oil slick, I would recommend at this point that you change your pipette. Right? So there's one change of pipette. And then with your new pipette, go back into the PVP and lubricate your pipette, and then start moving these guys across one by one. So do number 1, move it across here, and then put it in its Eppendorf tube. Then do number 2, move it across, put it into its Eppendorf tube. 3 and 4, same way. Now, when you go into the Eppendorf tube, this is sort of the final part in the process, and this can be a little bit tricky. And in this picture right here, I'm showing you something that's good and I'm showing you something that's bad. The good part of what you're seeing in this process is that my hand is resting on the microscope stage, right? So my hand is kind of upside down and it's resting on the microscope stage because that allows me to have the greatest stability with my pipette right here, resting my hand right here. My hand is not sort of floating in space. It's actually anchored on the stage of the microscope. Now, that's good. The hand is anchored. That's good. What's bad in this picture, and I hope you can see this, is that the pipette is touching the side of the Eppendorf tube before we get into the media. Right? You got to train yourself and you got to practice so that you can put your pipette in here without touching the side of the Eppendorf tube. Because the danger is you have your cells in here that have been nicely washed and you're trying to get them into the bottom of this Eppendorf tube, into this media. If you come in and you touch the side, your cells may come out just by capillarity. They may come out and end up on the side of the Eppendorf tube and not in the drop of buffer down here where you need them. So practice and be careful and get your technique down so that you never touch the side of the Eppendorf tube as you go in. Right? So over here, you can see that the pipette is cleanly in the media now. We're not touching the sides. This is what you want every time. And you should be able to visualize under the microscope that you see your cells going into the buffer right here. And then you know that the cells are in and that you did a perfect job in getting that DNA into this tube so that the genetics lab can do their analysis. So the tips here are: choose a small stripper tip, rinse in PVP as much as you can, don't touch the sides of the tube, visualize the cells going in, and rinse the pipette in PVP between samples. Now, many of the genetics companies will tell us that every time we load a sample in here that we should use a new pipette tip. These pipette tips are expensive. If you biopsy 10 or 12 embryos for a patient, you could go through a lot of these pipette tips. So in our lab here in San Francisco, we do a lot of embryo biopsy, and the expense of changing these pipette tips would be very significant. So the way we avoid having to change pipette tips all the time is we use PVP to rinse the tip between samples, and that allows us to avoid having any carryover. We actually do that for the biopsy pipette as well. The glass biopsy pipette, we also rinse that between biopsies because PVP is very good at helping you to sort of clean out the pipette between samples. All right, and then finally, the last thing that I want to tell you about, and this is sort of my last tip of the day, is that as embryologists or as IVF labs, we should know what the rate of no call is for the samples that we submit for analysis. So if you do an embryo biopsy and you send your little Eppendorf tube into the genetics lab and they do the analysis, then most of the time they should be coming back and giving you a result on that embryo. Right? But sometimes you will not get a result because they will say there was no DNA in the tube, or there wasn't enough DNA, or the DNA was degraded. Right? Now, as a lab, you should know what your no-call rate is. So we looked at our no-call rate in San Francisco, and as a group, our no-call rate was 1.5%. Right? Wasn't perfect. So for every 100 samples that we send in, 1.5% of them we do not get a result. Now, that's the lab as a whole. When we looked at the individual embryologists, and that's what you're looking at on the bar chart right here, you can see that there's actually quite a variation from embryologist to embryologist. Right? So embryologist A over here, when they did embryo biopsies and sent their samples in for analysis, they had a no-call rate of 2%. Now, if you look at the individual embryologists, you see there's quite a lot of variety here, a lot of variation. Embryologist C always got a result. They had a 0 no-call rate. When embryologist C submitted samples for analysis, they always got a result every time. That seems great, doesn't it? They never had any no-call samples. Whereas embryologist F over here had a no-call rate that was almost 3 times the lab average. So what's going on here? Well, your no-call rate will actually tell you if your biopsies are good. And when you look at this graph, and let's say I was to ask you, who is the best embryologist at doing embryo biopsies here? your tendency might be to think that it's embryologist C, right? But that's actually not the correct answer. The embryologists that are really good at doing biopsies are probably these guys right here, B, D, and E, right? Because when we went back and looked at these individuals and watched them doing biopsies, what we noticed was embryologist C was taking more cells during each biopsy than any of the other embryologists. They were taking such a big piece of the embryo that they always got a result when that sample was submitted for analysis, right? Their biopsies were too big. They were taking too many cells away. Now, in contrast to that, embryologist F over here was a new embryologist who had just learned how to do biopsy, and she was very tentative and very nervous about doing her biopsies. And whenever she got a cell or two cells, anything came off an embryo, she would stop and she would pull back and say, okay, I'm done, because she was nervous about it. So in contrast to embryologist C, who was taking too many cells, embryologist F, a lot of the time, was taking too few cells, right? And by looking at this data right here, we were able to figure all of this out, right? So we were able to go to this embryologist and say, you got to be a little more aggressive. You got to take a few more cells than you're taking. And she got there in the end, right? Embryologist G suffered from that problem a little bit too. And then embryologist C was taking so many cells that she always got a result. That's not good, right? You got to get it down to where, you're taking enough cells where you get a result almost all the time. Occasionally, less than 1% of the time, you don't. This is good right here. This is good. This is good, right? If you go to any genetics lab and you ask them what their no-call rate is for all the people that use their labs, they're probably going to tell you it's somewhere between 2 and 5%, right? So that's probably your target. As an embryologist, you're probably looking to hit a no-call rate of somewhere around 2 or 3%. And if you can hit a no-call rate of 2 or 3%, you're probably doing really nice biopsies. If your no-call rate is 0, you're taking too many cells. If your no-call rate is 5%, you're probably not taking enough cells, right? So you can use your no-call rate as a guide to tell you whether or not your biopsies are on par or not. All right, so we have reached the end, my friends. Thank you all for coming in and listening to our little webinar today. Today we talked about 3 subjects, right? We talked about incubation of embryos and equilibration of dishes and the use of nitrogen gas. Right? I'm not going to read through this summary right here. You can see that for yourself. We talked about vitrification and how important it is to set up your work area so that you can work quickly and efficiently. And we talked about lubricating your pipette to prevent yourself from having any problems, and about shrinking and re-expansion of cells, how we want to minimize that as much as we can. And then the last thing we talked about was micromanipulation. We talked about ICSI and anchoring your eggs so that they don't move during the ICSI procedure itself. And then getting your sperm to really clear the end of the pipette so that when you go home, you lock up your lab at the end of the day, that you put the sperm into those eggs, right? There was no doubt about it. The sperm shot in there and it was definitely inside the cytoplasm. And if you do that, you'll have fertilization rates of 85% or better. And then the last thing we talked about was blastocyst biopsy and the tricky procedure of getting little sticky pieces of trophectoderm into Eppendorf tubes. And you can use PVP to your advantage here to help you not lose cells during that tubing process. And then finally, always pay attention to your no-call rate, because that will tell you what the quality of your biopsies is. And with that, I'm going to say thank you to all of you for listening. My email address is very simple. It's Joe, J-O-E, @pacificfertility.com if anybody wants to reach out to me. But we are good and we are done. All right, so how do I stop sharing my screen now? So now we have some questions for the participants. We put together a couple of questions just to sort of see who was paying attention and who wasn't. So over on the right-hand side of your screen, I'm hoping that all of you out there can see our first question right here, right? And this is to do with the last topic that we talked about. So when I do an embryo biopsy in my lab, the rate of no call or no DNA in my samples or in our samples as a lab should be what? Can you guys see that? Can you all go down there and make a choice and then submit your answer? All right, I think we have 30 seconds for people to submit answers on this. You can think about this for a little bit. I can have a little bit of my coffee while you guys are doing that. All right, can we see the answers? Yeah, a lot of people didn't answer, huh? Maybe people are watching this on their cell phones or as they're working. So the answer we were looking for right here was 2 to 3%, right? You shouldn't have a no-call rate of 0. Less than 1%, not sure about that either. I think somewhere in the region of 2 to 3% is the sweet spot, because then you're not taking too many cells or you're not taking too little cells. All right, can we have the next question? We have a total of 3 questions, so we're 1 down and 2 to go. All right, next question. All right, that's the first question again. See if we can get the second question up here. All right, second question. You have 30 seconds. An embryo culture dish with media droplets under oil needs to be equilibrated in a CO2 incubator before embryos can be placed in the dish. If the dish was made using medium and oil from the refrigerator, what is the minimum time needed for equilibration? The minimum time. All right, 30 seconds is up. Let's see our answers. All right, pretty good. Everybody got this, 8 hours. So a lot of people didn't answer, but 8 hours is the correct answer. And really, you need a good 12 hours, but if you're in a pinch, maybe 8 hours is okay. All right, can we have our third and last question? Okay, we're back to the first question again. That's not it. All right, last question, maybe the hardest question of the bunch. In the IVF lab, where would you find sucrose? Which of these solutions would contain sucrose? And your timer has started. You have 30 seconds. All right, let's see how we did. Yes, vitrification solution. You guys did good. That is where we would find sucrose. The solutions that we use for vitrification, part of their job is to dehydrate the egg or the embryo before we vitrify. And by adding sucrose in the last step of the vitrification process in the VS, by adding sucrose, we make sure that we really sort of try to extract as much water as we can from the cell before it goes into the liquid nitrogen. Now, I saw one question come in on the chat on this very subject. Somebody on the chat, I didn't see whose name it was, but somebody on the chat asked, what happens if you leave your eggs or embryos in the vitrification solution for longer than 1 minute? Okay, longer than 1 minute. Well, there are many protocols out there. I've seen a lot of protocols where they say 90 seconds is the maximum time in the vitrification solution. And probably a lot of you will go slightly more than a minute, maybe as long as 90 seconds. The reason there's a time limit in the first place is because we're using cryoprotectants like DMSO in these solutions, and the amount of cryoprotectant in the vitrification solution is 30% by volume. It's a lot. And we're also using cryoprotectants that are good glass-forming agents that vitrify very easily. Right? So you have something like DMSO that forms the glass very readily, and you don't tend to see cryoprotectants like glycerol in vitrification solutions because glycerol does not form a glass very easily, or at least doesn't form a glass as easily as DMSO. So you're using cryoprotectants that are strong glass-forming agents, and the problem with that is that the better an individual compound will form a glass during the vitrification process, the more toxic it is to the cell, right? And because we have 30% by volume cryoprotectant in the vitrification solution, we really want to minimize the amount of time that the eggs or embryos spend in the VS. We really want to minimize it. Now, as I pointed out in the talk, cells are extremely stable when they're in that sort of shrunken condition, when they've lost their water and they've gone down to about 30 or 40% of their initial volume. In the VS, you do not need for them to re-expand. Once they've fully shrunken and they're washed through the vitrification solution, so there's no trace of the previous solution anymore, they are ready to go in the liquid nitrogen. There's no need to wait. The longer you leave them in there, it's just unnecessary exposure to high levels of cryoprotectants that they don't need. Get them onto your device, get them into the liquid nitrogen, and get them vitrified. Right? Now, if you leave them in there longer, it's possible that you're not going to see any obvious effects from this, but really, as you continue to vitrify and move forward, you can really refine your skills, you can hone your time, and you can get doing these procedures much quicker and much faster. And it saves you time too, and it saves the embryo or egg from sitting in the cryoprotectant solutions for longer than necessary. What was that question again? Do you recommend unlocking the cryolock? All right, so in the video I showed you where I did a warming, and I hope you could see this okay. You might have noticed that I took the cryolock out of the liquid nitrogen, and then I pulled the top off the cryolock, and then I went into the thaw solution. Right? Now there's two ways to do that. The way I did it is just one way, right? I took it out, I pulled off the top, and I dunked it into the thaw solution. The other way you can do it, and some people in my lab here in San Francisco do this, the other way you can do it is you can remove the cap from the cryolock under the liquid nitrogen. Right, so then you just take the cryolock out of the nitrogen and go directly into the thaw solution. If you do it that way, you got to be careful because you got to do this relatively quickly, but you don't want to do it so quickly that you take some liquid nitrogen and end up with liquid nitrogen in the thaw solution, right? I like taking the cryolock out of the liquid nitrogen and then briefly pausing to take the cap off and then going into the thaw solution. That sort of 1 second interval that it takes me to take the cap off, it allows any liquid nitrogen that's on the device to evaporate before I go into the thaw solution. If you get liquid nitrogen into the thaw solution, it's going to bubble, it's going to produce gas, it's going to obscure your view, and it's just going to make things a little more difficult for you than they need to be. So I like the concept where you take it out of the liquid nitrogen, quickly pull the cap off and go in, because that allows that liquid nitrogen to evaporate before you get the device into your thaw solution. But there are more ways than one to do this, so whatever works for you. All right, I think we're probably at the end of our time, Nicole. It looks like we're just have like 9:59 on my computer right here.
Yes. Thank you so much, Joe. We're going to conclude this session. I really appreciate you taking the time to present on these 3 tips. They're very valuable information that the embryologist can take away and use in their lab. So thank you so much, Joe. Thank you for all those that attended. There will be a recording that will be sent out within the next week, so you can rewatch it. So thank you again, and have a wonderful week, and stay safe and be well.